1 more minute. So actually what we can do is I'll start, it gives me a little bit extra time for the introduction because we need a lot of time for this. So,
No, you don't have to. You can just give me the floor. I can start talking.
no, I think you deserve the proper introduction, Ben. So Ben Yu is the Chancellor's Distinguished Professor and Class of 1936 Second Chair in the Departments of Statistics and EECS at Berkeley. I think what's really intriguing for me is that her work has leveraged all kinds of new computational developments. And she combines statistics with the computation with approaches from domain expertise in genomics and precision medicine. She's a member of the National Academy, the American Academy of Arts and Sciences. She's been president of the Institute of Mathematical Statistics. She's been a Guggenheim Fellow. She's been a Tukey Memorial Lecturer at the Bernoulli Society. She has an honorary doctorate from the University of Lausanne. She's on the editorial board of the Proceedings of the National Academy. It's really an honor and a pleasure to welcome you to Rutgers, Ben. We had hoped it would be in person, but this is absolutely great for us to have you.
Thank you, Fred, for the kind introduction and also for inviting me. And I see many old friends here. It's great to see or kind of virtually see you and hopefully meet a few new ones. So today I want to share kind of the work my group, my collaborator have been doing for the last goes back almost 10 years ago. Fred mentioned the Tukey lecture. That's where I started really getting on stability as a unifying principle for many things we do. And predictability was brought to the radar, of course, by machine learning was in statistics, but machine learning really made it big. And causality is kind of a new area for me. And I always thought stability is a minimum requirement for interpretation and causality, and predictability is the easiest to do reality check. So everything is coming together, and then I'll use the background of a biohub project to find genetic drivers of a heart disease called HCM, hypertrophic cardiomyopathy. So this is just put my framing on AI. Now everything's called AI, machine learning, statistics, data science, but at the heart, it's really this potential, like nuclear energy, as Bill Gates put it, but both promising and dangerous. That's what I see things, let's maximize the potential and minimize the dangers. And my world for the last 10, 15 years have been neuroscience, biomedical problems, and there's a lot of interesting problems that needs a lot of rigorous thinking, I think, than the average machine learning culture is used to. So I really think it's a great opportunity for statisticians, mathematicians, and other people who do slower science, and I think that's what's called for. And this is the background project. You can see it's really a collaboration of many teams. So most of the work I present mostly from my lab and you and Ashley's lab, the guy with the tie on the left, and from Stanford Medical School I'll present. And James was in his group now, he's doing something else. And my collaborator Ben Brown on top of UN. And I'll introduce my many team members from UN's group and my group when we go along. It's hard to remember, but it's really a team effort. So this is a proposal funded by Charles Zuckerberg BioHub 3 or 4 years ago. We really proposed a whole pipeline from using the UK Biobank data, from analytics recommendations to like a wet lab experiments in UN's lab. So we're actually pretty pleased that we actually achieved our goal. It was kind of pretty speculative when we wrote the proposal. So let me set the problem we try to solve, medical problem. There's something called HCM. Is if your left ventricular chamber, which depicted on the right, the walls get thicker, then your heart will gonna have a problem for normal functioning. And you can cause heart attacks and out of breath and fainting, all of that. So that's basically the kind of light brown, like a red area, just get thick walls and you have hard time pumping. So this is HCM, and Ewan's lab is really expert on this. It's about 1 in 1,500 in the US, people have this problem. And that's what I said, right? So we don't have to go all of that. And what the approach we take, the reason we working with you is our expertise for data science or statistical machine learning. And we have these 3 important pillars. That's the advanced diagram. And to really realize the vision Bill Gates articulated, and which I also agree, a trustworthy AI, we either maximize the promise, basically do kind of preventative data science, or we do damage control. So I'm concentrating on the preventative data science for trustworthy AI or data science. And if you want to do that, you really need to understand data science as a life cycle, not just somehow the data is in your computer, you want to run algorithms. That's part of it. But a lot of that part actually being very much taken over a lot by software, very well-made software that you can try many, many different algorithms. And a lot, I think the human energy should be put into the earlier part and other parts and interpretation, not just the down that node, which is modeling algorithms. And recognize all the human judgment calls we put into this data science cycle, which is not really sequential. Often you go back, you look at the data, you clean the data, and you realize that this algorithm has a problem with the way you clean the data because some missing value you interpreted wrong, and you might want to go back. And for problem formulation, you can formulate problem in many different ways. In the vague scientific problem here is that we want to find treatment eventually for HCM, but we first have to understand what might be driving HCM. So think really it's helpful, like old statistical quality control, think data science as a life cycle, as a whole process instead of just modeling stage. And I want to really hope you agree that this is just no-brainer. You want stability to the human judgment calls. Ron did some work, I did some work on the same data. We should end up with 2 completely different scientific conclusions. And that should not be acceptable. And we need a reality check. We might all both agreed on something, but completely both are illusionary, then that's not good either. So these 2 things, I hope it's no-brainer. We need to achieve this through quality control. And these 2 very basic requirements for trustworthy AI and data science. And that's why we start coining this new term, actually suggesting Tian Zhang from Columbia, vertical data science, use called 3 principle data science. Some of you might have earlier versions. It's really try to emphasize the quality and reliability and reproducibility information, and also through enrich technical language and bring in empirical evidence in a serious manner. Then, oh, I just did some p-value that works. And really seriously look at empirical evidence in the context of human decision and domain knowledge. So back to the HCM problem. We want to find genetic driver for HCM. You could say that, well, I want to help patients with HCM. I want to look at environmental factors. That's a different problem formulation, but it's completely legitimate, right? We want to do the genetic way. So the way we approach this, we'll have a causal question because we want causal drivers, not just association. So we're going to use biobank data, which is observational data. And then we use domain knowledge. We have to have an interval form, find the highly associated ones, and we use stability, push us further towards causality, not just predictability. And we discuss with humans, and then we also look at annotated database, and we'll do recommendation system. And then Ashley's lab did a knockoff experiment. Actually, to be precise, called knockdown experiment. They didn't do CRISPR-Cas9. It's really reduce silence the genes, not removing the genes. So it should be called knockdown experiments. So that's how we go for causality. We have recommendations, use predictability and stability and human knowledge. And then we discuss, and then we get that do knockdown experiments for confirmation. So for the rest of the talk, I will give you details about our PCS framework for vertical data science, more general framework. And there's a very nice paper right now. Somebody just called vertical spatial data science. So people already building on the framework for a specific area in data science, which the original paper didn't address. Just the example more like IID data. But all the high level principles are still there. Predictability, probability check, and how do you do data perturbation and algorithm perturbation. And then we use the idea stability to add to a very successful machine learning algorithm, random forest, and then make a new methodology development called iterative random forest. And we're going to use that to the HCM problem to do nonlinear model selection to reduce the dimensionality. And we have to do a lot more than just that. I'll tell you about to get appropriate data and binarization. Otherwise, the iterative random forest wouldn't work. And then experiments and find, we think, genetic drivers for both protective or harmful for this problem. And now I'll finish with some software we have built for people to use for the PCS type of analysis. So PCS, I used to give talks, some of you might have heard it, I have been giving for a while, like predictive computer stability. And the cooler name is vertical data science, means truthful. Vertical is an English word rarely used. In Spanish, it's used a lot. It means truthful. So this framework, as this picture try to depict, is really try to combine machine learning and statistics and build a bigger framework that streamlines and unifies both these ideas and push forward. And stability is not just about sample-to-sample variability or different methods as in robust statistics or local perturbation as in control theory. We use stability as a general conceptual framework, consider all the reasonable perturbations to the whole pipeline from problem formulation, linguistic stability to data visualization, data cleaning, all of that can be put under this stability. Like big conceptual principle, and the user design and argue with documentation why this is a reasonable perturbation to kind of stress test. And I believe stability is a minimum requirement for interpretation and also for causality. So documentation is a big part of the PCS framework, because for human judgment calls, sometimes just opinions and you want to write it down why you made the judgment calls. And the model is a mental construct. Reality is reality. They have no connection. They don't have connection because you put them together, because you run your model on the data. Doesn't mean you capture the right structure. So you have to make much more evidence to build a bridge, and with codes and judgment calls in like R Markdown or Jupyter Notebook. Otherwise, you have a pretty much a broken house. PCS also connects, I think, data science, both engineering and science, and predictability stability really embed the scientific principle prediction and replication. And computability in PCS is more than just scale up, faster computation and efficient use of memory space. It's also about how can we do data-inspired simulations, use so that we can learn things, use computation data or synthetic data as an argumented data to help us understand the method or even support the conclusions, even scientific conclusions, if you have a science-driven simulation. It's very much related to what people do use, like mechanistic model to simulate a robotic arm reach out to a bottle instead of having a graduate student reach a bottle and try to how to teach the robotic arm to reach the bottle and grab it. So it's in that spirit. And we have been also working with ER doctors from UCSF to stress existing decision rules. This is just from a collaborator. Well, he kind of reached out to me after I heard talking at UC medicine kind of conference. Didn't know Aaron before. He reached out to me because he thought this is the right framework he want to pursue. So we actually did stress test some clinical decision they already use in kind of a pediatric ER rooms. And I kind of embarked on this PCS framework through this stability work 10 years ago. At the time, there was a lot of report about non-reproducible biological experiments. And I thought Tukey was doing robustity. I was having problem with lasso, the interpretability, because you change the data, the lasso choose different predictions to interpret and put them together, and said that we need both stability right to do reasonable data perturbations and also model perturbations. And we use predictability also as a reality check. So stability, after I wrote that paper, I started talking to philosophers and said, this must be something people have thought about. Of course, it's so just common sense at the point to me. And my philosopher friend gave me this quote from Plato, saying for true opinions, as long as they remain, they find things, and all they do is good, but they're not willing to remain long, and they escape from man's mind, so that they're not worth much until one ties them down. So ties them down, I feel like it's really stability. That's why knowledge is priced higher than correct opinion, and knowledge differs from correct opinion in being tied down. So basically, knowledge, things want to take away, should be stable. Opinion can change. Now I think should be common sense. And stability is also a key to theory. Central theorem, if you look at Lindenberg swapping trick, it's really a stability, a perturbation show that the limit doesn't change. It's universal, and you can prove it through ODE after you assume the limit distribution satisfies something, use Taylor expansion. And then concentration inequality, basically you need stability conditions. And random matrix also have this both swapping, delete one column, delete one row. And in machine learning, you also have uniform stability. People have bounds on generalization. So I'm saying that not just empirically common sense, and mathematically it's also already in our work, that for the most central result for classical central limit theorem, but for modern limit result too. So it's both theory and practice, or practice and theory. And stability is a conceptual principle. It's not a cookie cutter to say you just do this and that. It's really a way to think. And if you want to really use it, you need to really articulate a reasonable perturbation in the data science lifecycle. For example, when you look at one-dimensional plot, violin plot versus box plot, gave me a very different impression. If you use different color, right? So that's what they call reasonable, because you could have chosen one over the other. And then you want your conclusion be unchanged to a certain degree to that reasonable perturbation, because you could have chosen one way or the other. So this all requires the user to articulate and make a case in the documentation why this is a reasonable perturbation. And then you have to argue why you should look at the difference in what way. So we did some COVID death prediction. We use 3 different metrics. If you're not sure, we use this absolute death count prediction. We take a square root of that. We also normalize it because relative change. So we actually want our method to work under 3 different metrics. So you can also have stability on top of the metric choice. And everything should be documented to argue for reasonableness. A lot of the reasonableness, you have to understand how the data is collected, especially when you do data perturbation, and include domain knowledge. And also keep in mind that if your work, whatever the decision rule, clinical decision will be used for another hospital, you don't want to randomly change your data for the test in your work, just do random split, right? You want to hospital split or group split. So the future is implicitly in P, in the PCS framework. You want to always keep in mind what you're going to use your data conclusion for. And that dictates a lot of things you do. You want the perturbation to be reflecting that, not just the data collection process, but the future possible perturbation. So for one thing, for data cleaning, that's why I'm advocating we should keep multiple copies of clean data, because in the future, similar data collected will be cleaned by a different person, most likely. And you have an extra level of uncertainty if you look at the whole process. And that needs to be taken into account instead of, if you don't do that, then you have underestimated uncertainty for how your result might turn out. And about causality, stability consideration actually everywhere in causal inference. If you're familiar with name and Ruby model, you have something called suitable. Suitable means that you have only one version of treatment. You randomize patient to control, say, painkiller, Voxy, or naproxen. That's some work I did, analyze the data. Then you want to make sure there's one version, the pill and gel form, and what time you take it, you basically, it doesn't make a difference. Otherwise, you have multiple treatments levels instead of just treatment or control. And there's no interference because somebody in the same family take a drug doesn't change the other person's outcome. So for infectious disease, if you want to look at vaccine, right, that's definitely doesn't satisfy. So a lot of the stability assumption in causal inference are actually assuming the perturbation doesn't change anything. So you have metric zero, metric value zero, conditionally, right? Invariance is very popular since the 40s in the from economics, and recently from the ETH group have been get a lot of more new attention to this through the Lasso work, but it's just a particular installation of the general principle already existing from the 40s from the economics literature. I recently discovered actually, like I was reading on the economics, I saw Abraham Ward mentioned, because he actually, before he went to Columbia, did some work in economics. And then randomization is really creating stability. You want the covariates to be balanced. So you want the outcome only depends on the treatment. Everything else becomes the same. So it's kind of creating similarity and stability. And propensity score is really assumed condition on the covariates, you know, things are independent again. And in causal, people actually do sensitivity analysis. That's definitely a stability analysis. So all of that is just very paramount and ubiquitous. And so the approach connected first to move from predictability and stability to PCS to causality is the thinking is that causality evidence should be a spectrum. It's not yes or no, right? It took the scientific field, like community, decades from the 1957 British doctors study on smoking and lung cancer until 1964 or 63. There was an NIH panel look at 23 or 25 observational study with no randomized trial to decide it's causal. We still don't know how exactly the physiology works. So it was a decision. There was enough evidence. So if you look at the writing of Nancy Cartwright, who is at UC San Diego, like more philosophical kind of causality, she would say that causality is about making actions. So you have enough evidence to act on it. And that's the practical meaning of causality, which I quite like, because that's usually when you believe something, you act on it, there's enough evidence. So the causality, the mechanistic model, like a Hooke's law, right? You put something there, and then the spring going to get longer. But it doesn't work 100% of the time, because if the spring is actually very old, the load is heavy, it might break it. You're not going to follow Hooke's law, right? So even there, it's not 100%, but it's very stable. It's very replicable relative to, say, FDA studies on a new drug. So we go for average effect or subgroup effect, but we're still assuming stability in causal inference. And so the idea is that we add stability to predictability. We do recommendation system. We come up with hypothesis generation and recommendation for experiment, and then we do experiment. So that's kind of the thinking go from predictive stability to causality. And stability consideration is really everywhere. As I mentioned, right? Problem formulation. You could formulate, I want to do environmental impact on HCM, not genetic environment. Data cleaning, I had run a class project last semester with 3 of my PhD groups, in my 3 teams of people look at the same data and public data with a doctor, UCS stuff for each team. They clean the data a different way. One team cleaned away 23% of data, and they reached different conclusions about what's important. And the metrics, some went for sensitivity and sensitivity, and what's the other one? Sensitivity and the false discovery type of thinking, and some people just went for predictability, susceptibility. And you have EDA choices, data argumentation choices, argument choices. So it's just everywhere you have a human judgment and stability metric. And you basically laid it out in the documentation, and you want the whole system doesn't break. So stability rating, you shake the whole system or replace each node by something different, which is just as reasonable. And then you make sure your conclusion don't change. And this is another example I often cite about a Harvard economic paper. And later people find that with different data exclusion, encoding error corrected, you reach different conclusions. So a lot of the first work was used to cite as high debt and GDP ratio is bad for growth. And many people cited for their austerity policy in UK and Europe, and later seem to fail to hold. So this is one very prominent example that big policies were made based on this, but actually was very sensitive to a few data points from New Zealand. And as I said, right, even in data partition, which is very common we do in statistics and machine learning these days, even do cross-validation. Do you do random split, group split, or time split? For time series, you should do time split because you're predicting the future, like COVID. It doesn't make sense to scramble up the data, remove the time structure. So that's why the future, the P, embeds the future should be always in your mind when you do your analysis, and to choose between these different choices. Or maybe there are other things you might do I didn't list. So this is one work we did for randomized trials for VOX. And we made our judgment calls. We made up different splits, and we had the test set. And the test set should be the best surrogate you can have for the future. So we first actually did a random split, later we did time split, and luckily the subgroups we found didn't make that much difference. So we basically found subgroup, this VOX, which $1 billion investment by Merck has been removed by the market, from the market, seemed to be pretty good for a subgroup of patients. Because there was cardiovascular negative impact for the general population, but for subgroups, we can avoid that. And we have many statisticians here, and same problem. I think you guys probably going to use different. I've asked statistics department many times. Usually the answer was always like, we probably do different things. We choose different algorithm for the same problem. So that's another perturbation. And climate scientists already were ahead of us. They already did that. You look at the global temperature rise, you see exactly, you have 9 climate models. Kind of based on the same sign, but different judgment calls on the granularity and on how they take into account observational data. So you have different, it's actually more informative. The global temperature at the end of the century is predicted from 1.5 to 5.5. It's a lot more informative than taking one model. The problem with one model is also give you the sense, oh, that's going to happen, deterministic. But this just know that we are not completely sure. There's a PCS inference part in the paper, which I won't have time to go to. We actually give perturbation intervals to take into account all this reasonable perturbation and just collect the intervals and use data in prediction setting to give the coverage, very much in the same spirit as a conformal analysis. You have the interval and you want to assess the coverage. So people often ask, there are too many perturbations, definitely. So you can favor different paths if you have good evidence. You basically be a little Bayesian now, have more weight on this path, and you write a documentation about it to argue why. And you cannot really do all possible paths because it's computation extensive, but you should actually take a few paths, at least 2 paths, I would say, to see whether the results are different. And documentation to say why you did, maybe computation. Often a lot of data processing for genomic data, R and seq, I've been told, have 2 standard normalization packages. You probably want to process your data with both and then run down, see whether you get the same conclusions. At least I think keep 2 for each, for data cleaning is a good idea. So now back to the HCM, right? So we have HCM labels from UK Biobank. We basically have found no signal. We use the SNPs data, do our RF, nothing, just 50-50. So the breakthrough really came when Weston, whose photo there was a graduate in Ewan's group, extracted new phenotype from the MRI heart image, which called left ventricular mass, the whole area, 3D. And that's a regression problem. Again, we don't know why it's signal, because regression, we don't know the signal noise level. We don't know why it's good or not. So we actually binarize it to take, there are other things, why not other phenotype, which we pursue now, but we haven't. So we binarized to the top 20% and the bottom 20%, made into binary balance problems. We know if we hit a 50% benchmark, there's signal. This is very weak signal. There's a lot of environmental factors we're not taking. This is adults. This is people from 50 to 75, not young kids. If you have data, same data for the young kids, signal will be a lot more clear. And we did stability analysis. We did top 15 and top 25, and results pretty similar. So this is a formulation. We end up formulating as a binary classification, not a regression problem. First, we start with binary classification with HCM labeled. There are a lot of people are HCM are now labeled, so a lot of false negatives. So we saw no signal. And we got the continuous phenotype, LVM, and then we banalize to force a clearer signal, and also have a benchmark to know we're getting something. We get consistent 55%. So pretty weak, but it's amazing the experiments that could show that we found things. So here's the data you have been hearing about. We have a 30K white British unrelated population. The relative have been processed out. This is a standard data set. And each person has 50 million imputed SNPs. So they only measure about 80,000 of them. And then they use genetic understanding and imputed the rest. So that's what we got. And the number of integers is a number of minor alleles at the SNP, single nucleotide polymorphism positions. And then we have now binarized high level, low level, positive, negative cases. So first is dimensionality. And that's our contribution to the proposal in 2018 was this work called iterative random forest. And what we did is a development adding stability. This is one of the motivating examples for us to really end up with the PCS framework, which actually the paper was with Carr, and then Basu is now assistant professor at Cornell, and Ben is a long-term collaborator in the department, also at IBR, and Carl, my former student, now at the UCSF as a postdoc. So what we really try to do is pattern discovery. So computer scientists, computer vision doing the simpler problem. You know you're looking for a cat, it's pattern recognition. Pattern discovery is that you want to find a pattern and prove it's meaningful. So you actually don't know what it looks like. So there's a lot of interior combinatorial searches. And we know that if we follow the statistical work of polynomial pairwise interaction, polynomial doesn't quite work. And also you have the main effect, second order and third, you just go up very quickly and you also run out of computing power. You can not only do second order, because we know from Drosophila that there are false order interactions. This is how Drosophila decide on the segmentation. Segmentation is like our spine. The body plan get laid out through first the segmentation, and you see like 4 genes, famous genes are acting to make this happen, the positioning of the straps. So we want to go at least to false order. And we have so many different new methods now, but I still think the domain knowledge is very, very important, because you want the analytical match between the statistical machine learning method with the science. So there's something for the French flag model in Drosophila and the other biology, saying that the biomolecules interact after certain threshold, because there's so much noise and Brownian motions going on in the cell at the molecular level that nothing's deterministic. There's always a stochastic noise, and the abundance is important. If you only one such a thing, nothing will happen. You need abundance, so that's the threshold in behavior. And that's a good match for decision trees. So that's why we like decision trees, also random forest, have been producing very good predictive results. But we couldn't interpret it because it's very unstable. So people before us already did that. If two genes or SNPs fell on the same path, people say that's interaction in one of the trees, one of the paths in the random forest. Random forest, basically you do decision trees, and then you do many of them by randomly selecting the genes. And then you get different trees and you just kind of somehow aggregate it. So we take the output of the trees, we don't take the average, and then we look at the path. We want to make it stabilize. So the way we did the stabilization, that's random forest, is add stability. So this was a motivating example for the PCS framework. It's saying that we're going to do self-dimensionality reduction. So we're going to look at the importance measures after random forest for each genes. And then we're going to downsample the ones who don't look very important. We never delete anyone, but they just get sample less instead of uniform sampling. And we also have random intersection trees to find intersection quickly. And we have an after loop of bagging the whole thing for stability. So the output is a stability score. So that's what you see. So on the left, we start with random forest, we downsample, and then we look for overlap for the many different trees. And you can see that we did 3 iterations of random forest with downsampling of unimportant ones of the genes, and then we basically don't lose. This is on test any predictive accuracy. On the right, you see the stability score, we just cut it at 0.5. This is actually, yeah. And then the red ones are pairwise interactions, and the blue ones are 3-order interactions. 3-order interactions, we don't have experimental techniques quite yet, which is the frontier to test experimentally 3-way. But for the 2-way interactions, we found 20. And we submitted the paper, and the referee said, you know, why we should trust the interaction you find. And then we went back to the literature because we have a very good collaborator, Sue Seneca, and who basically gave us the references. We talked to her and she said, oh, you look good, but we actually didn't get a reference until we submitted the paper. So actually 20% or 80% of pairwise red interactions already in the literature, people already done experiments before the high throughput available. So this was a really promising, make the methodology promising. We did a lot of simulations here to use the Boolean models. And now we're doing a follow-up paper instead of just they're on the same path. We also say that the split is positive and negative. So we call the sign interaction or SURF. We're putting on the UCSF genome browser track for other Drosophila biologists to use for the enhancer problem. We're just about ready to submit the paper, have been going on for a few years. And I usually like to come back to theory. I was very theoretical when I started, but now the way I do theory is I do empirical evidence to see something works, and now I do theoretical analysis because I want to see whether there's any insight. And also a lot of people will want to see guarantees. So the other way around, I don't trust theory. I don't want to do theory just because, you know, for the theory's sake, but I want theory to help practice. So here in this paper, accepted by PNS, we led by my former postdoc, Merle Burr, and former student, Yu Wang, and also joined by another student, Xiao Li. We really couldn't quite analyze each random forest because with too many randomness in the algorithm, but we made up a theoretical version of it. So what we did is that we look at the same random forest output, and we just look at the path. Before, if there's a split, we just take them and we use the average. So what we did is we put stability in the jump. When you do the split, it has to have a lower bound, have enough jump. So the split is pretty stable. Then we have a theoretical version called the LS find. And we have a new theoretical model for people to analyze. It's a linear combination Boolean interaction, which is more sensible for biological applications and better fit to analyze model selection consistencies. Lipschitz type of condition, I just feel like it's not the right match for a lot of the high dimensional problem, because in high dimension, when I have Lipschitz condition, usually things probably not very interesting in all these different dimensions. Now we can prove consistent model selection consistency. However, in simulation studies, when you don't have enough data or something, don't have the right model, each random forest outperforms the theoretical version, because theoretical version is very, very delicate. And each random forest will have a lot of empirical evidence. And we do have a relaxed criteria. So if you find, so you have a 3-way interaction, you find 2 of them, that's good enough. That's how science works. You don't always find everything. So use that metric, iterate favor definitely is better. So recall that for HCM, we need dimensionality reduction. That's what we're going to do, use iterate random forest. And then we do domain knowledge discussion and the database, and we do knockdown experiment. It's not knockoff. So the pipeline, we do iterate random forest. We actually first use the traditional GWAS, use bot LMM to first reduce to about top 1,000. And then we do iterate random forest. And already we binarize the data. Because iterate random forest is not fast to run on millions of parameters, millions of features. So we have to do marginal selection. And then we rank the genes. We have a new stability-driven criteria. Even we split at the SNP level, but when we aggregate it, we map to the genes. So we have another level of stability analysis, and we have a gene level score. And that's how we rank the gene interactions and genes. So that's the people. Tiffany will be on the market next year, so watch out for her. She's wonderful, and to show already in industry, in volume finance firm, and Abby is a third-year student, just transferred to study from physics. So those are the, just look at T10, TTN is already known gene to impact LVM. And we find CCDC 141, which is almost next to TTN. We find other these genes, and IGF-1R is a growth factor. You can see that probably related to the sizes. So things are reasonable what we find. And we also find 3-way interactions. And we actually, when we now work with the experts, we worry about confirmation bias. So we have a protocol I'll call domain expert opinion solicitation with negative controls. We give them 3 lists. 1, what we really like from the model, 1 middle range, not so good, and the last thread, like random findings. And our collaborators pass our test. So far, all my collaborators that are great scientists, they haven't failed. So we don't want them just to confirm with us. We give them false ones to see whether they are able to tell them apart. And then we, under the help of Chad, who is a young cardiologist fellow, work with us more closely, and with Tiffany and myself and the other people, we basically look at the database. So the database only tells marginal importance, and they seem reasonable, right? So this is just sanity check. All the top genes seems to be not, but you can see it's also confirmation bias. So that's why it's not the proof. I should call it knockdown experiment. And then we went to the lab, Ewan's lab, and they already have the stem cell or iPSC cells for this cardiomyocyte. This is one of the 4 types of heart cells, like they basically the basic units for the muscle to work. And we did a gene knockdown. So it's not CRISPR, many will probably heard of it. It's really silence the genes. Okay, it's called siRNA transfection. And the sample were prepared by the undergraduate student, and then the working with the postdoc Chen. Jordan is the undergraduate. And Chen was educated at MIT with mechanical engineering, working as a postdoc in Ewan's lab. She has been the last few months our main collaborator. She designed this sorting machine to sort the cells, and we can count. So, and she also built software to do segmentation and the top ones. And based on our feedback, she actually redid the experiment. So we really became an integrated team to work together very closely. I took my team before pandemic, worked with, visit Stanford, even though closed for a whole week, just met everybody in the lab. And that was 2 years ago now. So this at least been going on for 2 years. So now what you see is what the cells moving under the microscope. And then the blue dots are the ones that the package is found. You can see that we miss some things, could be fragments. And then the top outlet are the bigger ones. And then we discover some humongous cells get, counted multiple times. So we'll have to remove that. So it's really helpful. They didn't have time to do this. We did it to watch and see what could go wrong. But amazingly, Chanru's actually segmentation was pretty good. So it's more the input became, she improved her flow rate and all of that to get things much better separated. And so improvement of the algorithm through the mechanical design, not through a software. And that's her pipeline. And we actually vetted her pipeline by manually looking at things. And we were very impressed that actually it was a pretty good. And my postdoc, Anna Kenny, who will also be on the market next year, watch out for her, she's also great. And my current student, Omar, did a lot of hard work to really just humanly look at this and try to do the marking and compare with the software. And we actually said her software does pretty well. So we couldn't really improve. We stress tested, and then she did change other experiments setup. And this is what we discovered, right? Some of the, you see this big cell, got, other things are zooming by. This cell got counted many times, and it's probably multiple cells. And we were able to try to remove such cells. So here's a particular, so there are two cell lines. I'll only show you one experiment. They have the healthy line and also HCM line. And the knockdown genes, we knocked down two, the CCDC and the growth factor. CCDC is next to TETON, which is a known kind of heart gene. And you can do all kinds of trimmings, but we have over 10,000 data points. And everything will be significant. So I don't really trust that, but we might need to do that for the reference. So I really want to see actually is the QQ plot. And you can see that definitely there is the scrambled quantiles relative to the knockdown. Scramble is already cell. So you can see that it's below the diagonal line. So the knockdowns, you knock down both genes, there's actually protective effect because the cell size got smaller. So that conjecture said when the cell gets big, you're more likely to have HCM. So of course there's a jump, right? So we can only look at the cellular level. So that cells get big, it's bad. So this is just a summary of the pipeline that we got. Cell size worked, Chanru, Omar, and Anna worked closely with Chanru, and we diagnosed the clumps and fragment problem. There's something else I didn't show you is that we also went in and tried to assess the measurement error. So they actually had bees with fixed size and to see for different lines they get different sizes. So we make sure that that's below what the differences we see. So we also did that. And we also removed cells that just got repeated. And then we formally want to do trim min test, but I really like the QQ plot a lot better because with 10,000, everything's significant. I don't trust the p-value. So right now we're working on a paper jointly with the UN lab to write up this work. So the last part, just say that I started with PCS stability, and then we showed you a scientific collaboration. And we have since then have built a vertical flow called vFlow software on my website. And we also have another package being made called SimChef. Remember I said C, computability in PCS, it also means have the good data inspired simulation. So design RF, we actually simulated many data, synthetic data from the additive Boolean models to help us to tune the iterandom forest, because we only had an enhancer data. We have a splice data we don't want to touch at all. So only one data set. We use the structure, the feature structure of the enhancer. We use the Boolean model, similar a lot more data set, and we know the truth to really give us confidence how to tune the iterandom forest. So that's what I hope the SimChef would do is in R. I think from what I understand that we really, it's quite improvement from the existing packages we have in the literature. And the vertical flow is by the team undergraduate student and my CS student, Chandon, who did wonderful work graduating, Jim Duncan, who is a biostat student, also co-lead the project, and IB again. So we really make prediction screening and stability analysis, because when you do stability analysis, if you don't do good parallelization, it just takes too much time. So make everything simple from the user point of view and reproducible and computation efficient. And we also make intuitive capacities to inspect, manipulate, visualize the pipeline. And we build hugely on existing packages from the CS literature, especially from Berkeley RISE Lab, Ray and ML Flow. And it's also agnostic to underlying ML framework. We're also working with Amazon's Glang and PyTorch, Scikit-learn, so that people can build into their environment more easily. And the SimChef is a cool name with this team of people. The person I mentioned is Corinne leading this with James and Tiffany. And Merle is already back to Germany, and Carl is using as a postdoc. So we provide powerful tools for evaluating methods, and with the documentation, we're also putting on the side. And we want strong, reliable, trustworthy data science. So this is also, so the first paper, vFlow, actually already been code reviewed, published in this journal called Journal of Open Source Software. And we have, I think, already 24K downloads, and we have 711 stars. I was told the stars are more trustworthy. And so it's being code reviewed already, and this one will be submitted. And this is an interface to show you that you can easily do this documentation, pretty handy manner. We're going to have like protocol documentation for other people to use. So to summarize, I'm a strong believer that data science and AI holds great promise, but we also have to make it trustworthy. And one way to make it trustworthy is to really vet the process. And you can interpret, but if the process is not trustworthy, I don't think just doing the interpretation at the end really going to convince me that's something we should use, especially in high stakes situation, public policy and medicine. And I showed you a particular development using the stability principle. You know, how do you do it? That's what I think the researcher will do in your problem. How do you add stability? It should be problem specific, and it can serve as a nonlinear model selection problem. And we use the RF with the HCM problem, give recommendations. We have to reformulate the problem and did experiments. And so we're very pleased that we only have the team. We worked for 3 years together. We have one paper related to PCS on IP3 episticity detection, and this paper. So only 2 papers, 3 years. We do slow science. And the PCS principles have been I've been teaching it for the last 10 years, and I'm finishing a book with my postdoc, Rebecca Barter, using PCS as a very accessible form of data science principles, and then introduce the other more traditional ones. So problem formulation, data cleaning, visualization, and all of that in the beginning and throughout the book on stability and reality check and communication. So here are the related papers, and here are the software. Thank you.
Well, thank you very much, Ben. Great talk.
Thank you.
Any questions?
I can share the slides with Fred, so I think it's much better. I can see people now instead of looking at my own slides. So any questions? Oh, hi, Chunhui.
I have one. So you mentioned that iterative random forests produce candidate genes.
Yeah.
And then you go to the domain experts.
Yeah.
And some of them are well known to them. Some of them are known to have more than just well known. Some are well known to have something to do with the heart. What percentage of the ones that you produced were not known to them?
Well, we only give them like the top few because they could only do 6 experiments, right? So I think CCDC was a surprise to them. Growth factor, the three important T10, they definitely knew, right? That was not a surprise. But CCDC, there seemed to be interaction with discovering. They didn't. They just thought CCDC just like T10, but that doesn't seem to be the case. And growth factor was not on the radar. So among the other ones, we didn't do experiments, so we wouldn't know. But the top three, I think one is known, two are not known. And I wouldn't go down our list very much at all. You know, we have very weak signal, right? We're getting 55% in like balanced case. So I changed my view because I always want to have good predictive accuracy, right? But now I learned that some problems just have really high noise. As long as you know their signal, this machine learning statistical method can still give them a leg up as a recommendation, not as confirmative analysis. So it turned out to be better than I thought. We were pretty much thought we have no hope after we did the HCM, just no signal. And then the phenotype working with Weston, who is a CS student from Stanford, really helped, right? He gave us a new phenotype. That's why you have a group people who can support you. And then we came up with the binarization, which related to my VOX result, because there we binarize everything. So I'm really borrowing ideas from different projects. Because binarization is denoising, I justify by saying that scientists always look at the obvious signals, right? So I said that makes sense. And then you run it, right? And then you see, you get more than 50% consistently. I felt like we saw signal. And in the continuous case, we have no benchmark. So this was exactly more than 2 years since we visited Stanford right before the pandemic. To get familiar with the people, and we met the student, we saw, I want to see what the capacity they have, what they could do.
No,
so that was a really good visit.
So anyone else? Other questions?
Let me ask a question.
Hi, Xiaohui.
Hi. Yeah, I certainly thanks for a very wonderful talk.
Thank you.
I certainly agree that you should really sort of perturb or do some simulation to check whether the conclusion is still stable. But there's some guideline that to what extent that this should be done, because if you keep on adding noise, let's say, or adding perturbation sooner or later,
yeah,
it will fail. So do you just report, say, to a certain level? I mean, if you perturb, then you can say to a certain level that the conclusion is going to change, or you simply say, okay, I'm satisfied with some predetermined level of perturbation, and then if the conclusion is the same, then I'll be happy. I will tell my client that's stable. So
stability, I kind of use it as a way to regenerate the data process, either about the future or so. That's my guideline, and that's qualitative. It's not quantitative. So you have to make a judgment call to say somehow the variability in the simulated is just as much as the existing data. But if you want to do stress test, then you do want to do more. And so that's, I'm leaving it as now as you argue and you do documentation. And then you hope you have something else like a follow-up experiment or something to really give you more evidence. I see it as really our work is one step in a series of future steps instead of one step conclusive. So it's like, do you have enough evidence to publish? That's kind of my question. Not saying that I made a conclusion.
I see.
Yeah, so I try to leave it qualitative because I don't have a quantitative thing. But my thinking is I want to regenerate with the data, almost like as the original data in terms of the variability. But if you want to do stress tests, I'll push further. And then how much further? I think your question is more relevant. How much? And I think the context, the domain knowledge, the discussion with the expert will help me to decide how far I want to push.
I see.
Yeah, so I rely a lot on domain knowledge.
Okay. Other questions?
May I
Oh,
ask
that's an excellent question, because maybe in the future we'll know better, but right now I don't feel like we can give a quantitative, like cookie cutter type of recommendation. There's more like principle, and you try to implement and do documentation. And the documentation, I think, help you to clear your mind. What do you try to do? But the guideline is try to reproduce the data generation process. So I just discussed with my pathology, urology collaborator UCSF this morning. There, we try to look at the big machine, deep learning models, and we do fine tuning for pathology report for structured data extraction. And we want to do some data perturbation for pathology report. So the question we discuss is, we want to keep some authentic writing. So we only can settle, we're going to do synonyms for different words. Even when you try to perturb two words together, you might completely make it not like a pathology report.
Yeah, I see.
Yeah, so we're very conservative. We're going to do synonyms.
Mm.
But I still think that's better than not doing it. So you have to keep the holistic structure. And a lot of the knockout experiments actually have this problem too. We did some other work. We knocked out a gene and the Drosophila just died. So you kind of got out of your natural distribution, because things are so dependent. But when you do this perturbation, you might not be respecting the dependencies, and then we'll have a problem. Yeah, so there we're only going to do synonym perturbation. Very conservative. And I think it's better than not doing it.
Yeah, also it's important to have a record of it. So, I mean,
Yeah.
that's.
And also put it to share, because I think we're just humans. If somebody's going to look over our shoulder, nobody's going to look at it. We're just more careful. It's just human nature. And for yourself, we also forget why we do things. So we're pretty big on documentation. So in my class, people have to do documentation in my group. And now we're going to do code checking too. Somebody else has to check the code. So bringing more and more these reproducible steps in the group. And then usually I borrow over to my class. It's a habit, right? If people just habit doing it, actually they get more done.
Thanks.
Thank you for the good questions.
Anybody else?
Yeah, may I ask a question here?
Hi.
Yes.
Hi, Professor Yu. So I'm Zijian, assistant professor at the statistics department here. So thank you very much for a very nice talk. So I just have a quick question. So you also mentioned in your talk that like people analyzing 25 observational studies to study whether the smoking is healthy or not. And so I'm wondering, say, like in your applications, so can you also utilize these multiple sources? So you got data from different sources and also give you actual evidence for this stability. Like say you got—
Yeah, that's kind of meta-analysis.
Mm-hmm.
I think people do meta-analysis, but I feel like people relying too much on some Gaussian models on top of meta-analysis. I'm definitely into like qualitative meta-analysis. That's what I would do. But quantitative meta-analysis, I've worried that people do have too much model assumption in there. But for HCM, I don't think there's much there.
You mean not much data or not much
studies to really say, yeah. In theory, yes. In practice, I think I think this is something for FDA, like expert panel to do. For myself, I don't want to do meta-analysis.
Sure.
Yeah, in theory, I don't dislike it, but personally, I would rather do, because you really, unless the people are really supportive, for me to do meta-analysis, I will have to ask so many questions they probably don't want to answer. So then if they don't answer, I don't know how to aggregate their results.
Thank you very much.
Thank you for the question. I'm not against it, but personally, I'd rather not do it. I think sometimes it has to be done as a political human policy decision. If I'm on a panel, I might have to do that, but I'd rather do this more like ground level work. I think suits me better because I want to know everything. And there you can't. Right? Yeah.
Thank you.
Thank you for the question.
Anybody else? Well, let me throw in one more. I mean, if I understood you correctly, when you first went to the domain experts, you actually fed in some findings that were false or misleading. Is that right? And then
What?
when you went to the domain experts,
Yeah.
and you gave them some top-seeded findings and middle range findings and so on, I thought I heard you say something about giving them some that were maybe not totally right.
No, I gave them the purposely wrong list for them to tell, so they don't just
Okay,
confirm with
that's
me.
what I thought I heard you say. Yeah. Could
Yeah.
you expand on that? You know, why?
Because there's something called confirmation bias, right? People tend to want to agree with you, they work with you. So want to make sure when they say this is work, it's not because they try to be nice to me, but they really know. So when you have a comparison, then they are much more sharper about what they really believe, what they don't believe. That's a well-known psychological phenomenon called confirmation bias.
Mhm.
So we try to give them a comparative, force them to really think harder, which one they really believe.
So what kind of response did you get for that?
They got the right one.
Okay.
No, you mean like whether they liked it? The good scientists, I tried with 2 or 3 group people, they didn't get offended or anything. They did it. Because some of them can think that you kind of not trusting them, but they are good scientists. They understand what I'm doing. So they're fine. And they got it right. They haven't failed.
But they don't do actual experiment on the bad suggestions, right? They just
No.
look at your list
Yeah,
and
yeah.
see if it makes sense or not.
Yeah, I just want to make sure they're not confirming because we gave them something they think, oh, that makes sense, because you can always come up with stories. So that means they could come up with better story with the right one, with the one we believe in from the analysis than the middle one and the random one. So they still came out with story, but at least differentiated it to 2 other ones.
Did you tell them what you were doing?
Yeah,
And did you tell them one group was not the right
they
result?
knew there was one. They knew there was one right group.
Thank you.
Of course, you could also tell them saying that whether all of them are wrong. That'd be interesting. We didn't do that. We could do that next time. Thank you. Yeah, we could do that. Just say we don't know. We give you 3 lists. Does anyone, right? So we did tell them one of them is the right one. We did tell them that.
Is there any literature about whether it's works better to tell people or not tell people?
I didn't look. I'm sure somebody might have done something about it, but this is homemade. Yeah, I didn't. I tend to do a lot of homemade things. So I need to talk to a psychologist. I'm sure I can. Yeah, I can talk to a psychologist to see if they have done. Yeah.
So, Professor Yu, first, thank you for your great talk. I learned a lot. So I don't have a question. I just want to say something. Because when I was still a student, actually, I regularly visited your website, try to steal some ideas. So actually, I remember you put a fable by the Han Fei on your website, the poor guy washing for the rabbit. So
Yeah.
I just checked, the fable was still there. So I think that fable echoes your today's talk very well. So that poor guy indeed has a, poor stability, maybe no causality, and lead to a very wrong, predictability. I think it's really matched
you.
your.
I forgot about that. I agree with you. Thank you. I forgot about that. Yeah. Yeah, it's true, right? We have causation definitely was wrongly, and then you have predictability. Well, causality is so related to stability, you can see it's there. Yeah, I agree with you. Thank you for reminding me that. I forgot about that. Yeah, it was 20 years ago, probably 15 years ago at least. Yeah. Great. Actually, I have another talk to give. So I will go, and it's great to see everyone. Thank you for inviting me, Fred.
We really appreciate
Thanks.
you joining us and for this great talk. We'll post it on the tripods.
Great.
And
And send me the link. I have some people who couldn't make it, so I'll send them the link. Yeah.
okay, that sounds great.
Thank you.
Thank you.
Thank
Thank
you.
you.
Well done.
Bye.
Thank you so much. Enjoy the weekend.
Bye.
Bye.
Thanks, Fred.
Bye.
Bye.